Good morning again. Wow, just trying to draw maximum attention. But let's focus on the nematodes and the traces that we can find of nematode DNA in ancient, sediments. This is work that I've done in close collaboration with Morten Rasmussen, James Hale, Regin Rønn, and Eske Willerslev from University of Copenhagen. And the system we've been focusing on is the so-called northern Pleistocene mammoth steppe. During the Pleistocene, massive ice-free land masses were home to iconic Ice Age megafauna such as the woolly mammoth, the woolly rhino, steppe bison, and so forth. Very sexy animals. And towards the end of the Pleistocene era, a massive mass extinction eradicated most of these mega herbivores from the planet. For several decades, paleoecologists have been discussing which kind of vegetation actually supported these large populations of herbivores. In the Arctic today, tundra is the predominant vegetation type. Tundra is dominated by dwarf shrubs, mosses, sedges, really boring stuff to eat if you're a herbivore. A lot of lignin, relatively high carbon to nitrogen ratios. It's a low productive system, and the palatability of this vegetation is low. So, some have argued that this type of vegetation could not have sustained these large herbivores. And it has alternatively been proposed that the Arctic mammoth steppe was to a much higher degree covered by what you could call Arctic steppe or tundra steppe. However, the paradox, you could say, is that today we hardly find any evidence of this vegetation type in the region or at all. And what kind of vegetation is it? Well, it's a more productive system dominated by forbs and grasses, which is perceived as a vegetation type of higher food quality than the tundra. The palatability is higher than for most of the tundra species. However, today, as I said, we hardly find this vegetation type. It's confined to, it's too early. Confined to southwest sloping, well-drained soils, and it's very patchily distributed. So, where do we look for evidence for vegetation of the past? Well, a nice thing about the Arctic is that it's cold, and much of the old soil beneath the Arctic systems is preserved in permafrost. So we have a lot of nice permafrost sediments that actually act as a big freezer, or you could say a hidden biological archive of the past. Sometimes even full-bodied mammals, such as this baby mammoth, drops out of the permafrost sediments. Come on. But we can also drill out sediment samples and look for traces of past vegetation, microscopic or even molecular traces. and combined with accurate or fairly accurate dating of the sediments, we can pinpoint these remnant biological remnants to points in time. Okay, so if we're interested in the vegetation of the past, the obvious choice is, of course, to look for traces of plants from the past. Traditionally, pollen has been analyzed. However, there are some drawbacks with pollen analysis. First of all, pollen analysis, or pollen spectra, are of course biased towards taxa with high pollen production. Further, pollen is often wind spread from quite great distances, so the pollen spectra at a given site doesn't necessarily give you much information about the local vegetation at the time point. And further, for some at least, pollen, the taxonomic resolution is not sufficiently high. From the permafrost sediments, we can actually extract microscopic plant fossils such as seeds, flower buds, leaf fragments, for instance. And we know that the macrofossils are locally derived, but again, we're hampered by inadequate taxonomic resolution in many instances. During the last decade, several publications have come out where traces of plant DNA has been extracted from permafrost sediments and sequenced to plant taxon. The plant DNA is locally derived, which is good, but again, when we're working with old DNA that has been sitting in the soil for hundreds or even thousands of years, it's highly degraded, so we're working with short fragments, which often restricts the taxonomic resolution. So my point here is that we already have paleo-environmental tools at hand, but they all have their advantages and their drawbacks. In order to get the maximum or the full picture, we need to combine different proxies. So I suggested to include soil nematode DNA as an additional proxy. And why do I claim that we can use nematodes? Well, first of all, nematodes are ubiquitous. So we find them in all environments. Further, they're abundant. Nematodes are the most abundant metazoa in soils. And of course, when we're really looking for traces of the past, we should start by looking for the most abundant taxa. And what we also know is that different vegetation types tend to host different nematode communities, so there might actually be potential for using them as indicator of past vegetation types. However, I didn't really have access to data on the two candidate vegetation types, you could say, certainly not for the steppe type. So first, I sampled modern nematode communities in tundra and steppe tundra sites in northern Canada and identified all the nematodes. And what came out of all this work was that two nematode families proved adequate as indicators of either tundra or steppe. So if we look at the step sites over here, this is the relative abundance of Teratocephalidae and Cephalobidae. And in the tundra sites, Teratocephalidae was very abundant, whereas I didn't find many Cephalobidae. On the other hand, at the steppe sites, Cephalobidae dominated, whereas Teratocephalidae were hardly present at the steppe sites. And both came out with high indicator values for their respective vegetation type, where the perfect indicator taxon would have an indicator value of 1. Okay. So I decided to move on using these two nematode families as indicators, and I was so fortunate to gain access to a lot of permafrost sediments that were sampled by a large consortium funded by the EU, the EcoChange project. They sampled sediment samples. No, it's not working anymore. Well, they sampled permafrost sediments throughout the Arctic region, from North America and throughout Siberia. And the sediments I worked with ranged in age between 6,700 years up to 39,000 years. So what I did was I constructed primers that were targeting very short DNA fragments of the two indicator families, the Cephalobidae and the Teratocephalidae, extracted DNA, amplified them with the primers, and subsequently we sequenced the amplicons on the 454 platform. So here are the results, and I combined them with the results from the EcoChange work on plant functional groups. So I hope this works. Yeah. If we start with the plants at the lower panel here, we have plant data that range back in time till 50,000 years before present and up to now. And in the in the Pleistocene period here, we see a long stable period where plant communities are dominated by forbs and grasses, whereas towards the very end of the Pleistocene and into the Holocene period, dwarf shrubs and trees start to appear in the plant DNA data. Looking at nematodes, I've compiled the data from the Pleistocene and Holocene sediments, and I present the percentage of samples that are positive for either the tundra indicator or the steppe indicator. And you can see the Cephalobidae, they are present in almost all sediment samples, whereas the tundra indicator, the Teratocephalidae, are hardly present in the Pleistocene record, but are found in half of the Holocene sediments. So the plant data indicates really a transition from a forb and graminoid-dominated plant community towards a more tundra-like system here at the transition from Pleistocene to Holocene, and the nematodes actually seem to follow this pattern. We see the tundra indicator when we move into the Holocene samples. So where does this take us? Well, I think in order to use the nematodes, we should definitely work for more taxa, and we should try to optimize our strategies, maybe enriching or going for target enrichment of environmental DNA samples to get rid of all the sequences that we're not really interested in, all the sequences that are not nematode sequences. what we could do is to look closer to both our environmental and also archaeological samples and specimens and try to see if we can actually track human, animal, or plant parasitic nematode DNA in such samples. And lastly, what I would find more interesting was actually to dig deeper into long-term nematode community dynamics in relation to changes in plant communities. So lastly, I would just like to thank Grant Sasula, John Davison, Tobias Mourier, and Jeppe Tander for help in the field and help with the analysis, and certainly the EcoChange project and the Carlsberg Foundation for funding. Thank you to you.
Thank you, Mette, and thank you particularly for putting up with the technical difficulties. We've got time for questions. If you could put your hand up clearly so that the people with the microphones can see one over there. You probably need the microphone.
Sorry? Not really convincingly, no. No, I didn't. I would, yeah, you could say that I would have preferred plant parasitic signals, definitely, but no. For the ancient part, I targeted the two bacterial feeders, so I couldn't get reliable data for the taxa that way.
Okay, any other questions? There's one down here.
How do you know that your nematode DNA was ancient? Couldn't they have been migrants from more recent times or even current times?
We have looked into this problem with potential DNA leaching down through the profile, you could say. And it seems that in these frozen sediments, it doesn't pose such a big problem. But what you could do is actually to check for the level of DNA damage in the sequences that you get out. I haven't done that, but that is certainly a possibility. So the higher damage level you have, the higher likelihood it is that it is actually ancient DNA.
Okay, one more question. Then we can have our tea break.
Thank you. I've studied early Waxlian paleosols in Belgium, trying to find which climate and which vegetation they had. Is there any chance that this would work on non-permafrost conditions? Would be really nice.
Would be. Let me tell you, it's really a hassle getting anything to work, even on permafrost samples. So there's been work on parasitic, or at least its sequences have come out from parasitic eggs, but there you also, there you have actually the biological stuff there, and you extract DNA directly from that. But I would say it would be great if you could get it out from temperate or even tropical sediments, and I think people are really working hard on it, but even on the frozen sites, it's really, really difficult. And I've looked at very many negative PCR results.
Okay, can I just ask you one more question? What's the limit in age? How far back can you go?
Actually, I haven't checked up on that. It seems to increase the limiting age all the time, and I'm really not sure how far we can go now, but the first ancient sediments that came out from Siberia, they went back to like 300 and 400,000 years. They actually got sequences that old.
Okay, that's good. Well, thank you, Mette.